I will present the PhD project of one of the co-tutel students we were talking about earlier. We will talk about Priscilla Carnavali's project on splenic function in adhesion molecules in sickle cell disease, which is a co-tutel program between the University of Paris and the University of São Paulo. The French supervisor is Professor Jacques Lyon, and the Brazilian supervisor is Professor Dimas Covas. And we have a Capis Coffee Cube scholarship for this. The number of circulating red blood cells result from the balance between the production of these cells in the bone marrow and the clearance of aging cells by phagocytosis in the spleen. So the production of red cells is called erythropoiesis, which takes place in the bone marrow, and accomplishes the development and maturation of red blood cells from early stages, since proerythroblasts, into the mature state of erythrocytes. And in the bone marrow, there are specialized niches for erythropoiesis called erythroblastic islands that comprises one macrophage and around 5 to 30 erythroblasts that are going maturation from early forms, nucleated early forms, into enucleation and young reticulocytes. The erythroblastic islands were first described in 1958 by Bessis et al., and it is essential for erythroblastic proliferation, maturation, and enucleation, and it regulates in some part the proliferation and death of red cells, so-called red cell homeostasis. And within the erythroblastic islands, the close relation of red blood cells with macrophage cells occurs via adhesion molecules. The first one was the erythroblast macrophage protein, and others described afterwards. And in the final stage, there is a migration probably of erythroblastic islands towards the sinusoids to permit the release of young reticulocytes that lost their adhesion, some of their adhesion molecules, to be released into circulation. And but the spleen also has macrophages that interact very closely to red blood cells, and those spleen macrophages are responsible for the removal of immune complexes, pathogens from red blood cell surface, the clearance of unwanted damage from red blood cell membranes, such as Auger bodies, the rest of chromatin nuclei and stuff. And there's a possible role that we're going to study in removing adhesion molecules from reticulocytes to facilitate the maturation, the final step in maturation of red cells. So, as you can see in this picture, we have many contacts between the spleen macrophage and red cells through many adhesion molecules, many different adhesion molecules. And under severe anemic stress, like in hemolytic anemias, one of them is sickle cell disease, erythroblastic islands can form within the spleen. So this dual function highlights the pivotal role of the spleen in red blood cell homeostasis, production and clearance. I won't talk much about sickle cell disease pathophysiology because Professor Jacques Lyon just did that. So, but we know that this abnormal hemoglobin polymerizes upon deoxygenation, causing cellular alterations, leading to the worsening of hemolytic anemia and vasocclusion. That will lead to acute and chronic organ damage, virtually in all organs of the body. And the abnormal adhesion molecules in sickle cell disease, and especially the adhesion to the endothelium, plays a major role in triggering this vasocclusion process. And especially under stress, that we can see in patients during the winter period. They get many respiratory diseases, virals and infections, and that culminates and causes worsening of vaso-occlusion phenomena that we can tell because there is a high income of patients into the emergency room. So stress plays a role in triggering adhesion molecules, activating those molecules, such as lubricant, which is very, it's a molecule very studied by the French group. And so under stress, those adhesion molecules are more activated and can trigger vaso-occlusion more efficiently. And there is another hemolytic anemia, hereditary hemolytic anemia, called hereditary spherocytosis. It's characterized by small red blood cells with high hemoglobin concentration, with a spherical shape called spherocytes, with a shorter half-life and increased splenic restriction, and which the clinical picture is very variable too, from asymptomatic patients to severe cases of hemolytic anemia. And the defect, the hereditary defect in the red blood cell membrane, causes an instability of the cytoskeleton and loss of cell surface, causing anomalous exposure of phosphatidylserine, which is a marker of cell apoptosis. And this molecule is recognized by the macrophages in the spleen, leading to splenic macrophage phagocytosis of those cells. So, the hypothesis of our project: in sickle cell disease, increased red blood cell adhesion to the endothelium results mainly from the release to the circulation of young reticulocytes expressing adhesion molecules. So, besides its role in clearing altered agent and sickle red blood cells in the peripheral blood, the spleen plays an important role in the maturation of young reticulocytes, as it contributes to the decrease in the expression of adhesion molecules in the surface of these cells. So, without the spleen, which happens in the majority of sickle cell patients after 1 to 2 years old, the adherent reticulocytes will circulate more and favor vaso-occlusion events in chronic hemolytic anemias. And our objectives of this study will be to evaluate the splenic role in relation to the reticulocyte adhesive phenotype in sickle cell patients before and after the loss of splenic function that occurs very early in life, and to compare the reticulocyte adhesive phenotype in sickle cell disease patients with hereditary spherocytosis, with and without the spleen. And in order to do that, we will study 20 patients with the splenic function, 10 sickle cell patients and 10 patients with spherocytosis, and 20 patients without the splenic function, 10 sickle cell patients and 10 hereditary spherocytosis patients. And we will analyze 3 parameters. The splenic function by analyzing high bodies and pit counting. We will analyze the adhesion molecules on the cellular surface of red cells by flow cytometry. We have a great panel of markers for adhesion molecules, including lubricant. And we will study the red blood cell adhesion properties to the endothelial cells by using a dynamic adhesion platform, which is another part of the collaboration between the French and the Brazilians, because this is a technology transfer. Also, they will help us in putting a machine here and starting working with this adhesion platform. We will start with sickle cell disease, but we will be able to use this dynamic adhesion platform to study other diseases and cells. Well, what have we done so far? Well, First of all, we need the approval for of our local committee that we got it in July of this year. And we perform standardization of the methods and techniques because Haut-Joli bodies and pit counting is technique. It's a labor technique that depends much of the expertise of the technician. And Priscilla never done this before. So now she's starting to learn how to look at red blood cells and look for Haut-Joli bodies and pit counts. You will say that's not easy. Pit counting, it's the visualization of red blood cell membrane vacuoles that appears as cells age. So once the cells have, red blood cells, for instance, have any damage on the membrane surface, the macrophage from the spleen will try to clean this red cell so it can become normal again to circulate. If the damage is too much, the cell will be phagocyted. So once we have many vacuoles in circulating cells, it's a sign that the spleen is not functioning well, because the function of the spleen, one of them, is to clear those vacuoles from the cells. And once we have pit counts over 3.5% are indicative of loss of splenic filtration function in sickle cell disease. So now she studied 4 patients, and all of them have plus way above 3.5% of pit counting. So those patients lost their splenic function. And Haut-Joli bodies, it's the rest of chromatin nuclei. We can find basophilic nuclear remnants. I don't know if you can see very well, but we have a picture here. And it also suggests a decline in splenic function with values above 0.3 per 10 cube red blood cells. So as we can see, all the 4 sickle cell patients that we studied, we got way above this threshold. So signaling that all of them have lost their splenic function by this method too. And now we are validating the freezing and thawing of our samples, because part of this work will be done in Paris, because it's a co-tutel program. So we will do part in here and part in France. And in order to study the samples in France, we will have to freeze them. So we are in this part now. Priscilla is trying to validate freezing process. And she will talk to some of the French researchers to settle a good protocol to do this. So after standardization and validation of the freezing and, of the samples will be sent to France, and Priscilla will evaluate the red blood cells' adhesion properties using the adhesion platform. And then she will learn how to manage the equipment, and we already bought one, but it's not here yet because importation takes a long time in Brazil. And for next year, we will have this platform here in Brazil, so we can start performing some studies here too. So we will be a technologic transfer. And in Brazil, she will perform the immunophenotyping of these adhesion molecules by flow cytometry, because we were waiting some antibodies and some beads that are necessary to start the project that just arrived. So now she has everything to really start her project. And what we expect to see, this is speculation, but we expect to see higher expression of adhesion molecules on sickle cell reticulocytes after the loss of splenic function, showing the splenic role in the clearance of such molecules. And we expect to see a similar adhesion molecule expression on reticulocytes of patients with hereditary spherocytosis after splenectomy in relation to the expression in sickle cell reticulocytes. Well, actually, we want to see in both diseases that after the loss of splenic function, we have more adhesion reticulocytes in circulation, showing that the release of the circulation of such reticulocytes is due to the chronic hemolysis and the loss of splenic function, not only for disease-related. And I would like to thank the two teams involved in this project. The one from Hemocentral, the head of this project is Dr. Dimas. Priscilla, this is your work. So, and we were going to work together because the patients that she's studying are under my supervision, so I will help her. And Daniele is the microscope girl, so she will help us with the microscope analysis. Maristela is the cell girl, so we're gonna need her to help us freezing and thawing and taking care of the sample so it can be analyzed. And Simone is the research girl, so she will help us with all the research issues that come along. And on the French side, we have Professor Jacques Elion, which is the head of this program, Wassim El-Nemer, and Claudine Lapourmeroli. They are pretty much involved in the Adhesion platform, so they will help and teach Priscilla how to use the Adhesion platform and transfer its technology to us. And Caroline Levanquin and Yves Collin, they work a lot with adhesion molecules on surface of red cells through flow cytometry, so they will help us a lot too. And once again, this project is sponsored by Capis Coffee Cube, and it belongs to the collaboration between the University of São Paulo and the French University. Thank you very much for your attention.